our 8th edition of our Grease Thief Thursday webinar series. Today's presentation is going to be done by Marissa Brandt. Marissa has been with MRG since 2019. She is our senior chemist, and rightfully so, as she holds a BS degree in biochemistry from Millersville University of Pennsylvania. She works in the lab analyzing grease and oil samples, preparing data for customers, helping us hold those conversations with the customers to interpret that data and what we're seeing. She also manages the quality program. She's a member of the American Chemical Society and of the ASTM as well. So I will let Marissa take it over from here to talk about Vapor Pro and measuring moisture within greases. So thanks, and everyone enjoy Marissa.
Hi, Trick. Thanks for that wonderful introduction. As Trick said, I'm Marissa. I'm going to be talking about moisture analysis using the Vapor Pro XL. So why do we check for moisture? Why do we care about moisture? So moisture contamination can form rust particles. It can lead to corrosion, bacteria growth, fretting, oxidation, loss of film strength and lubricant. It can lead to viscosity or consistency issues. It can also affect the acidity or the alkalinity of a lubricant. And all of that can lead to equipment failures or effect on our products produced. So here on your screen, you see two different images. The one on your right is a microscopic image of metal. And then on your left, we see a microscopic image of metal that has been affected with moisture and pH. And we can see that the effects of moisture can definitely affect metal surfaces, and we also do not want that in our equipment. Here's also another extreme picture of how moisture can affect different metal surfaces. As you see here, we have seen strong oxidation caused by moisture contamination. So just with that in mind, this is why we want to look for moisture in greases. So why are we using the Vapor Pro? We love it because it's chemical free. We don't have any expensive glassware. It's a great alternative to Carl Fisher. It's easy to set up and it's really simple to use. And you'll see a demonstration of this later on. So how does the Vapor Pro work? So first we have our sensor down here. And then we also have our needle, which is a 2-chamber needle. So up here we have our flow regulator, which regulates the flow from our dry air source, which in MRG labs we use nitrogen for that. We then have our flow sensor, which measures the flow regulation. We also have a heating component. Right here, this is our vial, and this is a representation of sample in the vial. We have a control board. Then when the sample goes in, it is hit with heating, and the sample is heated. Then we have our dry air source coming in. Again, in MRG, we use nitrogen. We have our dry air source coming in. And as our dry air source is coming in, we have moist air coming out of the second chamber using that needle. And that is going to go hit our sensor. As it hits our sensor, the control board then reads the flow sensor and reads the polymer sensor. And then that gives us our answer in either percent water, parts per million, or micrograms of water. And this is the sensor very close up. We have different layers. We have a thermoset polymer, then we have the porous platinum layer, we have another thermoset polymer, then we have another platinum layer, and that all sits on top silicone. And this is just used to see how much water is being counted. So some key features we like about the Vapor Pro is it's a sensor-based technology, as shown earlier. So it gives significant fewer interferences than Carl Fisher. We also have a much lower consumable cost than Carl Fisher. And then in terms of data transfer, we have a USB option. You can also do an optional web service as well. Other things is you're able to monitor temperature, rate, sensors, all of that. So over here to your right, you see that over here is our rate graph. So this line in blue is going to be your results in ppm. And over here we have our rate, and we see our rate goes up, it peaks, and then it slowly comes back down. Down at the bottom, we have a temperature graph. So this red line right here is temperature. In this current scenario, the temperature was constant, but you can also have an option to do step change temperature if you would like to program that into your Vapor Pro. And right here, you can see the different readings that we can see is like flow, pressure, things like that. So some other key specifications for the Vapor Pro is you have a wide range to test. So we can test anywhere from 25 degrees Celsius to 300 degrees Celsius. Here at MRG, we range from about 100 degrees Celsius to about 175, depending on the test we are running. It also has a great wide range of moisture detection. So we can go anywhere from 10 ppms all the way up to 100 ppms, which would just be water. We also have a great variance in sample size. So we can do anything as small as 0.01 grams, anywhere up to 8 grams. So you can see here, before you test any sample, the Vapor Pro makes you enter the weight to do its calculation. So you can see that over here, that you're able to enter the weight of every sample. So this is great with sample size because you can use small samples or you can use big samples. So if you have a customer with a very small amount of sample that can only be tested, we can take just a pea size of that and put that in the Vapor Pro and get a very accurate and very accurate reading on that. And the results can be in percent moisture, ppm, or micrograms of water. Some other great things is it can store up to 250 different test programs. Here at MRG, we don't use that many. We only use about grease, oil, chiller, hydraulics, and glycol. The Vapor Pro also stores up to your last 1,000 test results. And of that 1,000 test results, it stores 1,000 graphs. So every time you get a result in Vapor Pro, it also gives you a graph. And so it will store your last 1,000 test results and your last 100 graphs, which is great to go back if you need a clarification of something. It also is able to perform statistical analysis like mean and standard deviation, which can be super helpful. And it's very adjustable. So you're able to adjust your ending criteria. Do you want it to end on your rate, on your time, on time then rate? So it's a very customizable instrument, which is great. So why do we use the Vapor Pro? It's a great insight in service grease contamination. One of my favorite reasons to use the Vapor Pro is you can start the analysis and then you can have the test run, and while it's running on the Vapor Pro, you can go walk away. It's kind of a set and forget it. You put it in, you start the test, and then you can walk away and do other analysis while it's running. And then when it's done, it will beep and the sample will come out of the instrument. It also allows us to measure moisture contamination for any different type of lubricant by just simply changing the test program as shown earlier. It's super easy to calibrate, prep samples, and clean up, and I'll do a demonstration of that, of the prep and the cleanup. So it's super easy to do that, and it gives you very accurate and reliable results. And in here you can see that all you need to run the Vapor Pro is a simple bottle, a cap, and then a substrate of grease if you're running a grease test. So Vapor Pro has many other applications besides just lubricants. It can also be used in plastic and pharmaceuticals, and there are many ASTM standards listed for those as well. And there's also different Carl Fisher ASTMs. And anywhere where a Carl Fisher ASTM is being used, you can substitute or do a modified version using the Vapor Pro. So it's very versatile in terms of changing between Carl Fisher and Vapor Pro. So are there any questions at this point before I do a live demonstration? So here behind me, we have the Vapor Pro. In a previous Grease Thief Thursday, we saw we extruded a grease on a substrate via ASTM 7918. And so that is right here. So as soon as a grease is extruded here at MRG Labs, what we'll do is we will take one of these substrates off. Easy enough. And then we will just put it in a Vapor Pro moisture cap. It can be a little bit tricky, but you're just going to put it right in. Just be able to put it right into the cap. And then you have a cap with a septum in it, and you're just going to put that right on. And that was our prep. So unfortunately, we're lucky enough that the grease leaf analyzer did all the prep for us. So that was all the prep for Vapor Pro. It's now ready to go in the instrument. So we're just going to lift this up right here, and the cap, there is a little ridge right there that's just going to sit right on the Vapor Pro. So we're just going to put that right in there, and it's just sitting there, ready to be tested. We're going to put the lid down, and for this, we're going to choose the grease method, and now it's asking me to enter the weight. So I already measured this weight, but the weight is 0.25 grams because that's what a substrate is, and I also already measured it via a scale. So then, once I enter the weight, it's telling me, okay, we're ready for your sample. Go ahead and click test. So I'm going to click test, and it's going to say insert your vial now. So I'm going to go and insert my vial, which was already done, and the lid is down. So now we're going to test it. And the Vapor Pro then brings in, I don't know if you heard it, but it's now going to bring in that vial into the instrument. So the vial is no longer there. It's now in the instrument being ran. And the first 30 seconds of the Vapor Pro is purging. So it's getting out all the ambient moisture either in the vial or in the air, so that's not affecting our results. So it's really great that the first couple seconds is already purging and getting out, so when we know our results are accurate because it's only measuring what's in the sample. So then this is again a set and forget it. So this is going to run, and I can go ahead and walk away. If I wanted to, I could sit here and monitor it. I can pull up a graph and see what the graph is going to look like at that time. I can also go back and I can also look at sensors and see what my sensors are reading while it's running, if I would like to, or I can just wait for it to be done. So that's pretty much how the Vapor Pro works here. Back here you see we have our nitrogen source coming in and it's running. Are there any questions or anything at all I can answer?
Yes, Marissa. If you could just explain or talk a little bit about the septums that you already mentioned and how, if they can be reused or not in the vials.
Great question, Richard. Yes, so the septums can be reused. You do not have to buy new septums every time. The septums just put a little hole at the top where the needle goes in. And as long as you are able not to see through them, as long as you're able to still have a good seal on them, they are still be able to be used. So you do not have to change your septums every time. Like you have to clean them, but you do not have to buy new ones every time. They are able to be reused as long as they are clean and dried. Same with caps are able to be reused, and even our moisture vials themselves, you do not have to throw away that glassware. You can simply clean them and dry them, and you're able to reuse them as well. So again, a very green, efficient way where we're not using any harsh chemicals, where it's very efficient. We're not buying new glassware every time. So it's very green as well. Great question, Richard. Any other questions?
Another question that we didn't see you weigh the sample. So how did you know that there was 0.25? How would you know that the weight of the sample?
Great question. Yes, I previously did weigh that sample on a scale. We do know that when a grease is extruded, about 0.25 grams go on every substrate, but I always like to double check, so I always weigh it as well and then put it into our instrument. And there you go. We're lucky enough that the instrument ended in 2 minutes. It does give you the time, so it just beeps and it will pop out. And then there you go. So this is your grease. Do not touch the vial. It is very hot. So inside it is 105 degrees Celsius. So that means right now this glass is 175 degrees Celsius, and there are warnings that do not touch. It is very hot. So what we do is we just click this little button right here to eject the sample, and then now we're ready to run our other sample, and this can just sit here and cool. Are there any other questions or anything at all I'd be happy to answer?
The next question would be, do you have to calibrate or zero the instrument before each use?
You do not have to do it before each use. You do have to do it daily. The calibration is actually up here. So we're going to take a micro cap that is preset to a specific volume, and we're going to take that micro cap and then you just put in the instrument, and it has a calibration setting. So simple as putting this microcap in water, putting it into a vial, and testing it on the instrument for calibration. We do it daily. It does not be done after every single sample, though. Awesome. Thank you for that question. That's a great question. Any other questions?
Could move your camera, Marissa, and show us the graph that the result and the graph that we just got from the sample that we just ran?
Yeah, absolutely.
And then explain, because you talked earlier about the kind of the bell curve that we get where it goes up and down. Could you explain what would be a bad curve as well?
You guys able to see that okay? Okay, so we just got that result. So as you see, after every test, we can see a graph. So this yellow line right here, that's going to be our rate, and we see it went up and then it ended. So anything that does not look like that or look like a bell curve is not going to be a good test run. We also can look at the temperature. We can see, yep, the temperature was constant at 175 degrees Celsius, and make sure that the temperature didn't fluctuate or anything like that at all. We can look at the data, and we can look at what was ran. You can add your sample number, also add a lot number, a date, or anything like that. So that is, you can also print your results or send it elsewhere. So that is what the graph looks like. And we don't want to see, when we go to this graph of rate, we don't want to see it fluctuate. So we don't want to see it to go up and down. We want it to be nice and constant. It's typically going to be a bell curve or something like this. We don't want to see it go up and down and have a lot of noise. Any other questions I can
Bobby, to answer your question, we do have correlation data that we've done testing. We don't have it at the moment to share, but we can definitely get that as something parting to share between the data that we collected. It's gonna be the same, very similar response to what you're gonna get in Carl Fisher. Kind of like what Marissa pointed out, though, is that the level of effort is going to be a lot higher with Carl Fisher, with the expensive glassware and the chemicals that's involved. This process is just a little bit less involved as far as the effort to get the sample run. And then the thing that we enjoy is that, as Marissa mentioned, we first set a time. So our sample has a minimum time that it has to run for. And then part of our study at the beginning, when we started using this, was looking at the ending rate and finding the right ending rate for the material that you're testing. And then when we did our comparison, we did get very similar data to the Carl Fisher when we did our internal testing with the units. And that's something that we could share with you. We just don't have it right now to share. To answer David's question, the check standard of the accuracy of the instrument. So when Marissa held up the micro pipettes or the micro caps, that holds exactly 1,000 micrograms of moisture. So when we calibrate the instrument, you have to then get out the 1,000 micrograms plus or minus the tolerance of air, which in this case is 50 micrograms. So to pass calibration at the beginning of the day, when we run a microcap that's filled with 1,000 micrograms of water, we have to get between 950 and 1,050 micrograms of water, and it's very close to 1,000 very often when we're running this. Maybe, Marissa, real quick, we just show filling a micro cap, just so everybody can see the prep of that calibration as well.
Yeah, I can absolutely grab that. Let me just grab a beaker real quick and fill it with water, and I'd be happy to show that calibration process. So let me pop the camera back real quick. It's able to focus. Okay. So we have a beaker with water. It is labeled water. So we do have our beaker labeled with water, and we do have our micro caps. So my process is I just take our tweezers, and I'm going to take out a micro cap. I'm also going to need a vial. So I'm going to take out this is kind of hard to see because they're super small, but I'm going to take out a micro cap using the tweezers. And this is very hard to see, but once I put this in water, the vial will fill up with water very quickly. It's something that you learn to look for, but we're just going to do that. And it filled up that quickly with water. And so now we're going to tap off the excess. And we are going to put it into a moisture vial. And then we're going to grab our cap with our septum on it. We're just going to put it on. And now this is ready to go in the Vapor Pro. So I'm going to set this right here for now. And then I'll come and show you how we set it up on our program. You're able to see that. Apologize for the blur. So what we're going to do is we're going to click Programs. And there is a system calibration program that's already set into the Vapor Pro. So we are just going to click that. And then it's going to say Insert File. The file is already over there on the rack to be inserted. So we're going to click Test. And now it just pulls into the system and it goes. You can see the first 30 seconds are purging. As I mentioned earlier, we purge that to get any ambient water out of the air. So we are just measuring actually what is in the sample. And then that will run for about 5 minutes to run a system calibration. And while that's running, we'll see the results. Any other questions I can answer?
There's another good question is how often and do you have to clean the instrument?
Yeah, great question. So you do not necessarily have to clean the instrument. We have found that with grease, some of that stuff can clog the instrument. So for us internally, we do clean our instrument via the manual standards of using IPA, which is isopropyl alcohol, and we do clean our instrument about quarterly. And there are instructions on if you were to get a Vapor Pro manual on how to do that. But great question. Any other questions I can answer?
Marissa, this is Rich. Can you hear me?
I can hear you, Rich.
Okay. How do you store or handle the vials, the septums, and the caps so that they have no moisture contribution before you add the sample, right? So you just
Yes.
showed the microcapillary and you added that in with a known amount. So what are the steps that you take so that, it doesn't already have a certain amount of moisture in it and that gets counted as well?
Right. So what we do for emergency labs is when it's done, we take it out and we clean it. We do clean it, the vials specifically with soap and water, and we do clean the septums with soap and water, and we'll like just wash them. And then with the vials and caps, what we'll do is we will just take a paper towel or a lint-free wipe and clean them out. And we will just set those in the hood to dry overnight. And then the vials, we also put them on this rack, which is right behind me. And that's where our Vapor Pro vials go when they're done being washed. And they will sit there overnight. And then we will also put them in the oven as well, sometimes to dry as well if there's still moisture on them. You can also just take a lint-free wipe and just wipe out the inside to help dry faster and let that sit overnight. So normally any air, if you just let it air dry like any other dish, it should be pretty moisture-free. And then the purge at the beginning of the test also helps eliminate any of that extra moisture as well.
Okay, can you move your microphone down a little bit? I think it's getting covered up by your collar. And then a follow-up question to that would be, is there a way to check the background levels of the vials with the device itself? The background moisture level that might be in an empty vial.
Yeah, absolutely. So you can what you can do is you can run a system calibration with an empty vial, and you can run that into the instrument and see what readings you get. So if you get a super high reading, if you get 1,000 ppm of moisture, obviously your vial is not clean or dry. I would assume that at that point you would probably see some droplets, but you could definitely run a blank vial. You can just grab a blank vial and a blank cap, and just like that, it can go into the instrument, and you can go ahead and run that and check to make sure that it is clean and moisture-free and able to be ran.
Okay, and David Turner asked about storing them in a desiccator, and certainly that would help keep the background levels low so that just the ambient humidity where they're being stored doesn't accumulate a certain amount of moisture that becomes part of your measurement.
Right. But again, the purge at the beginning, it does a 30-second purge, which is meant to take away all that ambient moisture. So it's not, it doesn't straight start testing as soon as you put it in. As I showed with calibration with the other sample, it does take about 30 seconds or so to purge to get out all that ambient air. So it's not affecting the sample. Now, I would not recommend putting a completely wet vial in there. It is supposed to be dried and clean, and that's per the manual instructions. But if you are still worried about that, you can just test an empty vial.
So the purge is done preheating, so that there's a gas purge and then the heating begins, because otherwise you would be losing some of the moisture from the sample if you heated it, purged it, and you would be underreporting a little bit. So how does that timing work with the purge, and how does the purge know just to purge out the ambient moisture that might be along with the air at the beginning versus actually desiccating or starting to dry out the sample?
That, Rich, would be the advantage of being able to work with people like we have today. We have Doug on from Amatech, Arizona, and they're able to work with you, and they have a lot of experience with any material that could be looked at in the Vapor Pro, and they have different recommendations based on the material type as to if there is going to be a purge, how long the purge will be. So they're really good at helping people set up a program like that for the material. Doug was really helpful in getting us set up for the grease program, and through our study, we're finding that we can see the little bit of rate increase from that latent moisture when we cap the vial. You see that dive down to almost no moisture, and then when grease is heating up, it takes about 29 to 31 seconds for that moisture to be released starting. So our purge, for example, for grease is 30 seconds. At the end of the purge, you'll see that the moisture rate is down at almost zero. And then right as the test is starting, you will then see the rate increase, and the moisture is then being driven out of the sample and collected for the duration of the test.
So Richard, for the case that we're primarily using this device for, which is greases, it's important to be able to characterize what that what that initial program should be, what that time of purge should be, and really what the temperature and profile should be in order to optimize for measuring what's in greases. And that, of course, would be validated by introducing greases with known moisture levels to know that this is accurately representing that. To David Turner's question, which was, is there concern about water from the sample being carried out with the purge? That would be part of the way in which it's set up, the purge time, the purge cycle, and then the temperature of the testing of the grease to make sure that that doesn't happen, correct?
That is correct. And during our initial study that we did before we started using the Vapor Pro on the grease samples for customers, we did do that. And we were able to spike a grease sample with a known amount of moisture and then run the sample and then see that increase from the spiked sample versus the non-spiked sample. We were able to get that increase. I know from the study that I was going for like 1,000 ppm increase of moisture, and we were able to get, right around 1,000 ppm higher for that spike sample.
And actually, there was some initial negative balloting at ASTM on this, right? Because there was concerns about differences with different grease types, different polarities, and the ability of a given method in being representative of those different types. And then there was some follow-up testing, correct?
That is correct. We did do a wide range of greases, trying to cover, we definitely covered all of the popular and then some of the more obscure thickener types and base oil types to make sure that there wasn't going to be one that was acting extremely different or would need some kind of different setting. Correct.
Same note, and knowing that David, who asked the question, is a grease manufacturer. Certainly, a grease manufacturer could take a specific formulation and really optimize that for that particular grease to try to have a formula or a profile for the setup of this to really optimize on its behavior to really dial in on those moisture levels.
Yes, agreed.
Awesome. Thank you guys for that. Are there any other questions we can answer? Do we make sure we answered all the questions in the chat?
Yeah, I don't know that I heard the answer to this one, but Bobby asked, do you have correlation data with other tests, and can you show that data? And Richard, this might go back again to the work that you did for ASTM, where we did some preparations of first we dried greases, then we did spiking with known quantities of moisture, and then compared that to an oven method with Carl Fisher, correct?
Correct. Yes. So I said earlier, we don't have the data on hand to present right now, but we did collect, and we do have data comparing the Vapor Pro and the Carl Fisher, and they do correlate. It's just for us, the time, like Marissa mentioned, we can start a test and then we can walk away. Whereas needing somebody to, watch the Carl Fisher test to make sure that there's not an interference or something like that with the introduction of the sample from the oven coming over into the cell.
And Richard, was any of that data part of ASTM research reports?
Off the top of my head, I don't know. As Trig mentioned there in the comments, next week, please feel free to join us. Grease Thief Thursday, we're going to be talking about FTIR analysis for in-service grease samples. So kind of in the next step on a contamination path on this analysis talk will be FTIR. So thank you very much, everybody, and look forward to having you join us next week to talk about FTIR.
Thanks, everyone. Have a great day.